we placed one of the activity of LGD in the timetable, and we wanted to do the LGD, but LGD is the small, large group discussions where we used to post clinical scenarios and then we ask questions related to that. But we are not doing LGD. This is actually a revision session. So if you have any questions related to my lectures, I'm here to talk to. So guys, do you have any questions? My first lecture is nothing but the introduction to physiology. And the take-home message in the first lecture was very simple. Please try to understand that do not learn physiology in isolation. Learn physiology always in relation to clinical context, always in relation to the the knowledge needed for you to solve issues related to patients. If you do not understand, if you try to learn physiology in the context, in physiology only, and not relating with the clinical context, you will remain isolated and you will not understand physiology per se. So I have already given you two examples. One example related to the insulin receptor, and the other example related to one of the very novel innovation, which was sodium glucose co-transport present in the small intestine, in the GI. And because of this sodium glucose co-transport, you remember we have saved millions of children across the globe because of using oral rehydration salt. So please remember these two examples. And these two examples are important to understand and to relate that physiology is always should be learned in the context of, in clinical context. And I also, to emphasize my talk related to that physiology is the Nobel Prize of Physiology is not only alone physiology, it is the physiology and medicine. Because physiology is the basis of medicine, and I also reiterated that the 3 Ps are important if you really want to be a very known, the best physician of your time. So please focus on to physiology and pharmacology and pathology. And physiology always help you to understand the transformation of physiology and pathophysiology. And this transformation from normal to abnormal is the most important thing, where you can understand that how to interact and how to intervene into physiological context. The second part which I want to emphasize in my second lecture is I have already told you that all biological systems, most of the biological systems in our body are controlled by negative feedback. We have discussed examples of negative feedback. We have discussed examples in my lecture about the positive feedback. We have discussed about examples of feed-forward reaction. We have discussed how to calculate gain. So all these things are important. Please try to understand. You should know how to calculate the gain. Gain is important because gain tells you the degree of correctness and how efficient that system is. I gave you one example, if you remember. If the gain of a system, say for example, the gain of body temperature regulation is minus 33, and the gain of blood pressure is minus 3. Both are negative feedback mechanism, but so ignore the minus sign. 33 means this is the highly efficient system compared to blood pressure regulation system. So the more the value, more the numerical value, if you ignore the negative sign, the more the numerical value, more will be the effectiveness of that system. So that system will be more effective. So that's how you need to understand and you need to appreciate, okay? Can anybody, tell me what is the formula of gain? How you calculate gain? Anyone? So gain is equal to, if we take mathematically, correction over error. So how you calculate corrections? So correction is, in the numerator, correction is, yes please? Yes, say loudly. Final corrected value. Yes, final corrected value minus original value before correction over final corrected value minus original set point. Thank you very much. So this is the formula, please remember. So if you just, whatever the, things are given to you, if you calculate, follow this, you will understand that how to calculate, and based on to the value, ignore the negative sign because it's the negative feedback. So based on to these values, we can say that how efficient the control system are. Is there any question related to my lectures? Thank you. So I'll request Dr. Abdullah to be here. Dr. Abdullah is already here. Thank you, Doctor.
Assalamu alaikum. You guys tested yourself? Okay, how was it? Half and half? Good? Okay, that's good. So, let me do this first. Okay, the way the session will go is that I'll have the students come to here and answer the questions, okay? So I'll just pick randomly, or, we'll have volunteers. Just let me... Initially, this session was, I think it's called LGD, which stands for Learning Group Discussion. So last year, I think the TBL directors, they organized a session like the TBL, and the students had an open book exam. But for this year, I thought just to, give you multiple choice questions, familiarize yourself with the type of questions that you will get in the exam, and we'll discuss them. So this will be kind of revision of what you have been exposed to. And if anyone has any questions at the end, you are welcome, okay? So maybe we should start. Let me put this. Okay, any volunteer to do this question? It's very easy, isn't it? Any volunteer? Yep, yep. Can you come to here? Okay, so as you can see here, the question is giving you the concentrations of ions in the table. This is extracellular and this is intracellular, okay, for these ions. Now, the question is asking, which of the following, this describes the equilibrium potential for chloride, okay? So half the answers, remember in the exam, half the answer is in the question, okay? So you have to read the question very well. Here I'm asking about the equilibrium potential and chloride. So the key here is to use which equation? The Nernst equation, right? So can you calculate for us? Let me open Google. Okay, calculator. So we're just going to use the calculator. Okay, so go ahead. What's your name?
Arwa.
Okay, Arwa.
Okay, so to calculate it, we have
Speak
61 over the charge of the ion, which is -1.
Okay.
Okay, I'll write as -61.
Okay, very good.
Multiplied by log the concentration of the ion outside over the concentration of the ion inside.
Okay, so can you go there and see that?
So for chloride, it would be 125 over 5. Which will equal -80.
Very good, Arwa. Can you clap for her? So everyone got this? Huh? So why she put minus? Because of chloride. If it was calcium, it would be +2, right? And if it was sodium, +1. If it's potassium, +1, okay? So the answer is -85. So let me see if I can highlight this. Okay, good. So that's the equilibrium potential for chloride. So let me just write it down in here. So I'm just looking for Word, Microsoft Word. Okay, do we write in here? Okay. So the equilibrium potential for chloride is equal, in this example, is equal to -85 millivolts, okay? Now the next question is which of the following best describes the equilibrium potential for potassium, okay? Which, you know, this is very easy question, so I'll just do it as as Arwa showed us. So what you would do is similarly, okay? So it's gonna be 61 log concentrations of potassium in this example outside is 5 millimolar, okay? So it's 5 millimolar over 140, right? It's 5 over 140. What? Wait a minute. 61 times log 5, which is the extracellular, right? over 140. Yeah, so it's E, right? Okay, so the equilibrium potential for potassium is equal to negative 88 millivolt, which is in this case, it is E. So let me highlight this. Okay, so for the next question, who would like to do that? Yep, come here. Okay, so the question is asking the net driving force is greatest for which ion when the membrane potential of this cell is negative 80 millivolt. So the question is giving me what's the membrane potential. It is negative 85 millivolts, and it's asking which of the ions here in the table has the greatest driving force, okay? So in order to solve this question, what do we need to do? Can you tell us what's your name? Lena. So Lena's gonna tell us what we should do. Okay, so what we should do? Yeah.
Okay, so basically you should find out the 4 different equilibrium potentials for each ion alone. So we already found for potassium and chloride, but then we have to find for sodium and calcium. So after you find the...
So can you find them out?
Yeah, I have...
So, yeah, very good.
Should I...
So just write down here what's the equilibrium potentials for.
So after you do the calculations, we know that the E of K is going to be negative 88. Then we have the E of Cl, which is negative 85. Then we have the E of Na, which is going to be 66 after you do log and all the process. And then calcium, which is going to be 143.3. After you find all of these, you're going to subtract each one of them with the resting membrane potential.
Very good. So what's this equation?
So it would be, it's membrane potential minus the equilibrium potential of the ion. So in the case of potassium, for instance, we would do negative 88. Sorry.
85, the membrane potential.
Oh, yeah. Sorry. The membrane potential, the resting membrane potential is negative 85 minus, which turns into a plus in the case of potassium. So you do plus 88, which is equal to 3. And then you do that for each one of them. So negative 85 plus 85, which is equal to 0 for chloride. Then you have negative 85 minus 66 is equal to negative 151, I assume. And then negative 85 minus 143.3, which is going to be equal to, I think, 8.3. Negative, sorry. And then you'll see the differences, and obviously the biggest difference would be calcium going into the cell.
Very good. Excellent. Excellent. This is a... yeah. What's your question? No, so this is an exercise. There is no faster way. So this is an exercise for you to understand the concept. So the one that has the largest value is basically represent the one with the biggest driving force. And the signs here, when it's negative in case of calcium, it means that the movement is going to be inward, right? And if you see, for example, for sodium and calcium, the movement is going to be inward. For chloride, it's going to be 0. There would be no movement, okay? And for potassium, it would be outward because it's positive, okay? Then when you calculate the driving force. It's all clear, right? Yep. Does the mic work? Okay, so in this case, calcium is the right answer. Okay, so let me just highlight this. Okay. Doesn't work, right? Okay. Yep. Again? Yes, so exactly. So the sign tells you the direction of where the movement is going to be. And so here we are just asking about the ion with the greatest driving force. If the question was asking the ion with the greatest outward movement, then you would calculate and find out which one has the largest value with the plus sign, and if it's, a cation, then this would be if it's a plus sign for a cation, this would be the largest outward. If the case in chloride, it would be the negative sign with the largest value. Is this all clear? So half the answer is in the question. You guys have to read the net driving force is greatest. So I'm just asking about what is the greatest driving force. I'm not asking about the direction, okay? Is this all clear? The direction is whether the movement is inward or outward. So when you calculate the driving force for a cation, if the result comes with the plus sign, so it's +3 or something, this means that the movement is outward. If it's a negative, then it means that the movement is inward. In the case of anions, like chloride, it's the opposite. Plus means it's going to be an inward, and minus is going to be outward. Okay? Now, any volunteer for this question? Come on, you guys. I'll randomly pick. I have all the names in here. Any volunteer? Yep, come to the front. So let me get the attendance list so I can. Okay, now, so the question is, which of the following best describes the changes in the cell volume? So I'm interested in the cell volume that will occur, okay, when the red blood cells previously equilibrated in 280 milliosmolar solution of sodium chloride. So this means, what does this mean? That the inside of the cell has a 280 milliosmolar, in terms of osmolarity. This is what the cell has. The osmolarity is 200 milliosmolar, okay? Are placed in a solution of 140 millimolar sodium chloride containing 20 millimolar urea, which is a relatively large but permeant molecule. Okay, so I'm interested in this question in finding out what happens to the cell. Okay, so what's your name?
Hiba.
Hiba. So please, Hiba, tell us what we should do.
I just
Yeah, so how did you approach this question?
since the inside of the RBCs has more osmol than the outside, so the solution will move from the inside of the RBCs to the outside, so it will decrease, so it will shrink. But then because we have urea, I thought because it just passes the membrane, doesn't need specific channels, so it will return the RBCs to back to its origin.
Very good. But now why you assumed that the outside has a hyperosmolarity? Can you tell us? Because looking at the numbers here, so here the cell is 280 milliosmolar, right? And the number here of the solution in which the cells has been placed has a 140 millimolar sodium chloride containing 20 millimolar urea. Now looking at the numbers, if you ignore the millimolar, okay, it seems that the numbers is very low, right? But why you assumed it's a hyperosmolar solution? Why? Can you tell us? Yeah. Okay. Because you said the cell would shrink, right?
Yeah, shrink.
Why is that?
Because the red blood cell is more than outside.
Why it's more than outside?
280.
Okay, 280 is what?
Double the 140. Double. Yeah.
Okay, so it's double. Why you doubled it?
Because the numbers are given.
Okay, so let me tell you. So the idea is that, so what you answered is correct. The idea is that we have 140 millimolar sodium chloride, and when we find out the osmolarity, what do we need to do? Sodium chloride, does it dissociate?
Na plus
Yeah,
NaCl.
so it will dissociate into giving two osmotically active particles, the sodium and chloride. So when you calculate the osmolarity, it would be 280 milliosmol sodium chloride plus the 2 milliosmol urea. Urea wouldn't dissociate. So the overall osmolarity of the solution outside the cell would be how much?
300.
300, isn't it? So 300 milliosmol, which means it's hyperosmotic, isn't it? So initially this will make the cell shrink, right? Because water will be going from inside to the outside. But urea is penetrating, so it will penetrate the cell and it will, it will regulate the volume of the cell, okay? So initially, the volume, the solution will make the cell shrink, but then it will return to its original volume. Okay, is this all clear? Thank you, Hiba, that was good. Thank you. Okay, we all agree? Huh? Okay, so let's move to the next question here. Okay, so who's our warrior for this question? Okay, come. The question is equilibrium potentials for 3 unknown ions. So these are the ions Q minus, R plus, S plus are shown in the above figure. This is the figure. Note that ions S and R, so S and R are positively charged and that Q is negatively charged. Assume that the cell membrane is permeable to all 3 ions. Okay, let's assume that the membrane is permeable to all. And the cell has a resting membrane potential of negative 90 millivolt, okay? So this is the membrane potential. Which of the following best describe the net movement of the various ions across the cell membrane by passive diffusion? So I want to know in which direction the movement of each of these ions, okay? Yeah. So what's your name?
Yara.
Yara, okay. Yara, could you please tell us?
Okay, first of all, let's hope that I did it right. I would recommend to draw this question just to make it easier. So basically, the cell, we have negative 90, and we're trying to bring it to negative 75, positive 75. So let's start by Q, negative Q. So basically it's an ion we need to move it out of the cell to make it more positive. So we need to make the cell from negative 90 to negative 75. That's because the Q is a negative, an ion, basically. For the R, it's a positive, so it's a cation. And what we're gonna do, we're actually gonna put it inside the cell to make it more positive. And that goes as well for the S. So S is a cation as well, but the equilibrium potential is negative 96. So what we're gonna do is move it out of the cell to make it more negative.
Very good. Excellent. So which choice you chose?
E. So the answer is E.
Okay. Very good. So yeah, clap for it. Very good. Yeah, we all agree here. So the movement of Q is outward, okay, because this is an ion, and it has to move out in order to bring the membrane to its equilibrium potential, which is less negative than the membrane potential itself, okay? Whereas R is positive 75, and so the R has to move to the inside of the cell to reach its equilibrium potential, okay? Going from the negative 90 to reaching the positive 75. And for S, it is negative 96. It is an anion, it is a cation, so it has to move to the outside to make the inside more negative in order for the membrane to reach its equilibrium potential. Okay? So outward, inward, and outward. So it would be F, isn't it? Yeah? Not E, F. But you explained the concept right, okay? The S1 is outward because the equilibrium potential for S, which is same as potassium, it is negative 96 millivolt. And in order for the membrane to reach that equilibrium potential, this cation, which is positively charged, has to move out to make the inside more negative, okay? So it is gonna be F, okay? Yeah? You could do that as well. You could calculate the net driving force as well and, you know, check the signs. So that's another way, but like a faster way of finding out is just by imagining what would be happening for each ion, okay? So let me just highlight this. Okay. So F is the answer. Okay, so for this question, do we have a volunteer? Yes, please. Yeah, the girls are braver than the boys. Okay. Come on, guys. Here where you make the mistakes, not the exams, okay? I want you to all have a full mark. It is in my interest for you to do that, okay? So if you don't make mistakes here, you will suffer later, okay? Okay, so this question is basically asking you to match. So we have, you know, the different types of transport here, and to match it with the different scenarios in here, okay? So for the first one, the transport of sodium ions from the cytosol to the extracellular fluid. What's your name?
Aya.
Aya, could you please tell us?
So basically sodium is more concentrated outside. So if we want to take it from the less concentrated to the more concentrated, it's going to be by active transport.
Very good, excellent. So it's going to be by active transport.
Primary.
The second one, yeah?
Primary active transport.
So can you write down prime, like the number? So here it's primary active transport. So just write down D, which is the primary active. Very good.
Okay.
Now the next one.
This is glucose uptake, so it's, oh yeah. This is glucose uptake, so it is down its concentration gradient. So it should be by facilitated diffusion, because simple diffusion, basically it's impermeable. like it cannot
Is it
pass.
using ion channels? Or
Yeah, it's using carrier channels.
Okay, carrier-mediated proteins. So it's going to be here.
Sodium-dependent transport of calcium-2 positive from the cytosol to the extracellular fluid. Oh, okay. So this is a co-transport since we're moving the calcium and the calcium by having the sodium move down as concentration gradient.
Is it a co-transport or a counter-transport? Which
I'm
one?
not sure.
Okay, so this one, is it a co-transport or a counter-transport? Counter, because sodium, this is a secondary active transport. We all agree, right? Because it's going to take advantage of the down concentration gradient of sodium. So sodium would be moving to the inside while calcium is going to be expelled to the outside. Okay? So this is a counter transport. Calcium is going to move against its concentration gradient. So in this case, it's going to be E, right? You understand this?
Yeah. Can we repeat the same letter twice?
Okay. This one? This one?
Yeah.
So sodium-dependent transport of calcium, we know...
I meant, can we repeat the same letter twice? Because I feel like this is C2. This one.
Which one? Yeah.
9.
So 9 is the transport glucose from poor glucose environment into the intestine lumen, into an intestinal epithelial cell. Okay? So it's going from area of lower concentration of glucose into a rich glucose environment. So this is against its concentration gradient. But again, calcium is known to be moved by a secondary active transport, okay, not by a primary active transport. So in this case, it would be a co-transporter, okay, not a counter, because it's going to take advantage of the sodium moving to the inside, and calcium will move along to the inside against its concentration gradient. So it's going to be a co-transporter. It's all clear? Okay, so it's going to be D, okay?
For 9?
Yeah.
But it's only glucose.
Yeah, I know. Okay, so for 9, it's going to be transport of glucose. We know that glucose mainly moved by a facilitated diffusion. There are no active transporters to move glucose. So in this case, we definitely is going to be a secondary active transport. But glucose is going to move from outside where there is less glucose to the inside where there is more glucose. And so it will take advantage of the movement of sodium to the inside. Okay? So sodium is going to move down its concentration gradient. Along with this, it will transport glucose to the inside. Okay? So the direction is going to be the same. So it's a co-transporter. Okay?
Why don't we use the GLUT1 transporter?
GLUT1 is a type of the carrier-mediated protein, okay, which moves glucose down its concentration gradient, not against its concentration gradient.
Yeah, okay.
So for this, we would put E.
9.
Yeah.
Okay. And the movement of sodium ions into a nerve cell during the upstroke of an action potential. So again, sodium ions are more concentrated outside, and to have them enter into the cell, then this is against down their concentration gradient. So it should be facilitated diffusion.
By using which type of?
By using ion channels.
Very good.
So it's A. Aye.
Thank you, Aya. Very good. Very good. Any question regarding this? Simple diffusion is, again, it's about the terminology. Some textbook make simple diffusion, as a general passive diffusion, okay? This happens mainly, but here in Guyton, I think as well, they use simple diffusion for crossing of the molecules across the lipid bilayer, the movement of oxygen or carbon dioxide, where the molecules cross the membrane without the need of ion channels or, proteins mediated carrier. So they call that simple diffusion. But some textbook, they do exchangingly, use simple diffusion for passive diffusion, okay? But for now, like I taught you, use simple diffusion for movement of the molecules across the membrane without the need of anything. Facilitated is when using, proteins or ion channels, okay? Down its concentration gradient. So they all are part of the passive diffusion. So there is no need for energy. Yep. Okay, so the difference between co-transporter or sign port. So the counter-transport and co-transporter are secondary active transport. They take advantage of the primary active transport. The difference is that with the co-transport, the movement of both molecules are in the same direction, where sodium would be moving down its concentration gradient, and the other molecule is moving in the same direction but against its concentration gradient. You see? Whereas the counter transport would be same, but the direction is opposite. Sodium would be moving down its concentration gradient, with the other molecule is moving against its concentration gradient in the opposite direction, okay, to the outside, okay? Okay, so this question here, so any volunteer? Yes? So as you can see here, we have three traces. There is A, B, and C. And the question is asking trace E in the figure shows a typical action potential. So this one is for a action potential in the nerve. And B is a typical action potential from a cardiac cell. Okay, and the question is asking, what is responsible, increased conductance of which of the following ions is responsible for the plateau? You can see here we have in the cardiac cell, we have a plateau in the action potential. What's responsible for this? What's your name?
Huda.
Huda, could you please tell us? Please.
So for this, I chose the option C because voltage-gated slow calcium channels. Over here, the potassium is basically exiting the cell, but because the voltage-gated calcium channels are slow, at the same time, calcium is also entering the cell. So the basically net flow of calcium inside the cell and the net flow of potassium outside the cell is equal. So hence the the charge or the membrane potential doesn't change. And then once the calcium-gated channels do close, which is in this area, then the potassium is obviously, it keeps exiting the cell, so that's how the membrane potential decreases.
Very good.
This one as well?
So you just highlight this, so it's the voltage gated. So next question.
Should I read it? So which of the following perturbations in the nerve cell would account for the failure of the same stimulus to elicit an action potential in trace C? So
why this? So the question is asking why we don't see an action potential in C. If in an experiment, this is like in an experiment scenario. Sorry. So this is an experimental scenario. Blocking, for example, blocking of what channels would be responsible for the profile we see in C. Because in C, do we see an action potential? We don't see an action potential. So we must be interfering with which channels, okay?
So for this, I chose A because the voltage-gated sodium channels are the ones that are basically responsible for depolarizing the entire membrane. So if the voltage-gated sodium channels are closed, then the membrane can get depolarized, hence the action potential isn't stimulated.
Very good. Excellent. Very good. Thank you. You guys agree? So it's blocking of the voltage-gated sodium channels is what's responsible for the nerve not being able to generate an action potential. So if the membrane is not being depolarized to the threshold and generating an action potential, then, you know, the action potential wouldn't happen, okay? Now, so this question is from the cell structure and organelles. The liver is involved in detoxification of many poisons and drugs. Which of the following structures is primarily involved in this process and therefore abundant in the liver cell? Which one? B, the smooth endoplasmic reticulum, isn't it? Yeah, that's... Why not what? Do they really involve in the detoxification of the blood? No, this is the function. This is like a recall question. This is the function of the smooth endoplasmic reticulum, okay, which in the liver cell would be contributing into detoxification of the poisons, alcohol, etc. Okay, so let me just remove that. And okay. Okay, any volunteer for the next question? From the boys? Yep, yes please, come. Yep. Yellow shirt. Okay, so the question is, a patient has had a serious accident and lost a lot of blood. In an attempt to replenish body fluids, distilled water, you guys know distilled water? So this is like an ultra pure water, equal to the volume of blood lost, is transferred directly into one of his veins. What will be the most probable result of this transfusion? What's your name? Samhar. Samhar. Okay, Samhar.
For this question, I chose C. The patient red blood cells will swell because the blood fluid is hypotonic. Because we added distilled water to the blood fluid, so the distilled water have no concentration, so it will be hypotonic. And the red blood cells will have concentration, so the water will go from the distilled water to the red blood cells, so it will swell.
Okay, very good, Samhar. Can you clap for him? Very good. Yeah. We all had that? Yes. So the cell, the osmolarity of the cell is much higher than the fluid, okay? Because, you know, now we have diluted blood, right? And so the osmolarity of the red blood cell, okay? Sorry, I chose C, okay? So the osmolarity of the cell is much higher than outside, and so now water would be moving, or the extracellular fluid would be moving to the inside to make the cell swell, okay? Patient would swell because it's, yeah. Okay. So this one, an inhibitor of which of the following could be used to block the release of calcium from the endoplasmic reticulum? Okay, yes. What did you get? Yep, you with the pink shirt, yep. Very good. So it must be blocking the phospholipase C, isn't it? We all agree with that? Okay, I think this is straightforward. So basically, you remember the phospholipase pathway, okay, where we had, so if you look at all of these, you know, options, they are not really play a part in the phospholipase C. which will produce the DAG and the IB3, and then IB3 would be traveling to the, diffuse to the endoplasmic reticulum to bind to its receptor to release calcium. So when we are blocking the phospholipase C, then this would prevent the release of calcium, isn't it? Okay. Okay. Now this question, a drug designed to inhibit the response of cells to testosterone would almost certainly result in which of the following? What did you guys get? Yeah? C? Okay, why is that? Okay, very good. But the reason why is this? Because testosterone is a cholesterol-derived hormone, which will cross the lipid bilayer. Okay, remember this? The cholesterol-derived hormones, they can cross the lipid bilayer, and they will bind to a transcriptional regulator that will, translocate to the gene of interest in order to switch it on, okay? And so when we inhibit the response of the cells to testosterone, this will result in decrease in the transcriptional activity of certain genes, okay? So it's not going to interfere with the other pathways. Okay, so this question here. Okay, so can I have a volunteer here? Yes, please. Yeah. So the question is, a 56-year-old woman with severe muscle weakness, so she has a muscle weakness, is hospitalized. Blood analysis showed an elevated serum potassium concentration. So she is suffering from which condition? Hyperkalemia, right? Which of the following accounts for the muscle weakness? I'm asking about the muscle weakness. I'm not asking you about the resting membrane potential. So if I was asking you about the resting membrane potential, you would say it will make the resting membrane potential, depolarized, right? It will make it closer to the threshold. I'm asking why the hyperkalemia is causing the muscle weakness, okay? Because initially it should be causing higher excitability of the muscle cell, isn't it? But here it's resulting in weakness. What's your name?
Ismail.
Ismail. Go ahead, Ismail.
So I don't think hyperkalemia will cause any voltage-gated channels to open or close. Instead, hyperkalemia is going to increase the negative value of the resting membrane potential. In other words, making it more hyperpolarized. So at any graded potential, okay, no matter at any graded potential that could be, a strong stimulus, will actually not be enough to let to let the membrane potential be above the threshold, which is negative 55 millivolt. So I think my answer is A.
So Ismail, now we are talking about hyperkalemia or hypokalemia?
Hyper.
So with hyperkalemia, when there is an increased concentration of potassium, how does this affect the resting membrane potential? It will make it depolarized or hyperpolarized?
This will cause the resting membrane potential, it's going to make it hyperpolarized.
Hyper or depolarized? When we have higher concentrations of potassium outside, right?
Yeah.
How is this going to affect the resting membrane potential? It will make it less negative, right? Yeah, it will make it less negative, so it's depolarized. It will make it closer to the threshold.
Why?
Okay, because when we have higher concentrations of potassium, so when we have higher concentrations of potassium outside, how is this going to affect the resting membrane potential? It will make it depolarized or hyperpolarized? Depolarized, right? Because we're going to interfere with the concentrations inside, and now the equilibrium potential for potassium is going to be less negative, okay? And so this will make the resting membrane potential depolarized. But wouldn't this cause, more excitement of the muscle cell? Right? It will cause more excitement, but why does it result in a muscle weakness? Why? Yep, yeah, you come here.
Is it because sodium channels will be
No, it's
blocked?
good, it's good. Yeah, it's because the voltage-gated sodium channels would be blocked. But why is that? Why would the voltage-gated sodium channels would be blocked?
Because it's always going to overshoot if it's closer to the threshold.
But how is this going to contribute to their close?
Because potassium ions channels are open.
No. Yeah. Stay in here. It's good. It's good that you. It's good. Yeah. Because this is the purpose for creating this.
Like we said, when there's hyperkalemia, it's going to depolarize. Okay. When
So what's
there's
your name?
when we have hyperkalemia means that there's a higher concentration of potassium in the blood, in the extracellular fluid. And inside, relatively, it would be less. So potassium would diffuse into the cell. This would depolarize it. That's what means it, making it less negative, more towards the positive. Overall, the resting membrane potential would be depolarized, i.e. made more positive, higher up. So now, even if there was a really small stimulus, a sub-threshold stimulus, it would generate an action potential initially. So this depolarization, that we would reach the threshold faster at a small stimulus, and this would trigger the activation gate of the sodium channel to open. But this would also trigger the inactivation gate of the sodium channel to open. It's just that it responds slower. So we would have a depolarization, sodium would start entering the cell, and then we would have repolarization when potassium leaves. But in the first action potential we would have, it would be more excited. Our muscles would be more excited, we would be able to generate it. But in the next one, since there was no repolarization, it's going to make the inactivation gate of the sodium channel open again. Since we won't have the repolarization, we're not going to be able to generate another action potential. No more depolarization from the sodium channels can happen because both the activation gate and the inactivation gate of the sodium channel have to be open for flow of ions.
Very good. Very good. You understand this? Okay, very good. So you see the trick in this question? So I'm asking why this could cause the muscle weakness. Because initially it will make the muscle cell more excited, isn't it? But over time, because the hyperkalemia would interfere with the inactivation gate of the sodium channel, it will make the cell in refractory, okay? Because the inactivation gate would, require that the cell gets, repolarized very well. But if the cell stays in refractory, there wouldn't be no generation of the action potential, okay? And this is how it would lead to a muscle weakness, okay? So in this case, it would be the voltage-gated sodium channels are closed by depolarization, okay? So half the answer is in the question, okay? If I asked you about the resting membrane potential, then you would choose, that the resting membrane potential become less negative, okay? But I'm asking why it's causing a muscle weakness, okay? You all understand this? Okay, good. Let me put this. Okay. Okay, another volunteer for this, yeah? You are for the question after the one. Yeah? Okay, now, if we had, so let's look at the action potential here. Remember here, like if we bring the threshold, if the hyperkalemia is bringing the membrane closer to the threshold, right? It's going up, gradually over time. When there is, a constant or an increase in the concentrations of potassium outside. Initially, this would bring the membrane closer to the threshold, and when there is a stimulus, it will be more excited, right? because any minimal stimulus or graded potential would cause the action potential. But remember that we have here a repolarization phase, right? And we have an absolute refractory period here, which is marked by the closure of the inactivation gate. And the inactivation gate would not be opening until the membrane gets repolarized, isn't it? And so when there is high concentrations of potassium, this will make the cell in refractory because we are interfering with the voltage-gated sodium channels. Okay? Is this clear? Again? Yes, exactly. So it wouldn't be open to allow for a second action potential. Okay? All right. Is this all clear? Okay. Okay, so the next question is, the resting membrane of a cell is due to unequal distribution of charged particles across the membrane, okay? Which of the following pair of particles are most important in giving rise to the resting membrane potential? I'm interested in the particles that play a role in establishing the resting membrane potential. We know that the resting membrane potential has a value of what? Around negative, 70 or 90 millivolts, right? So what's your name?
My name is Lara.
Lara, okay.
Yes. So my answer was A, because in order for us to maintain a resting membrane potential, a sodium and potassium pump is needed. So two potassium ions would go to the inside, and 3 sodium would go to the inside in order to maintain a resting membrane potential.
Okay, very good. Okay, excellent. So basically, yeah, very good. So what establishes the resting membrane potential is firstly the high permeability of the membrane to potassium, the slight permeability of the membrane to sodium, and the active pump, which maintain the resting state to be at around the same value, for example, negative 90 millivolts. Okay, so it is the sodium and potassium that plays the significant part in establishing the resting membrane potential. Okay, is this all clear? Okay. The next question. What's your name? Come in here, yeah. So with the resting membrane potential, remember that all the ions, the unequal distributions of all the ions and the molecules, proteins, they do play a part in this unequal distribution, okay? But it is the sodium and potassium that establishes the resting membrane potential, okay? We know that proteins are more concentrated inside the cell, and proteins are negative. And this is why there is high negativity in the inside. But what gives rise to the value of the resting membrane potential of negative 90 millivolts is the sodium and potassium, okay?
Can you read the question?
Okay, so the question is the resting membrane of... Yeah? Oh. So a model cell with 3 different transporters, X, Y, and Z, and a resting membrane potential of negative 70 millivolt, okay? So this is the membrane potential. As shown in the above figure, consider that the concentration of sodium chloride and calcium is higher, okay? The outside, and the inside of the cell, potassium is higher inside, okay? Like, an irregular cell. What would happen to the intracellular environment? So I'm interested in what happens inside, okay? If a drug that inhibits the activity of transporter X was applied, so if we interfere with X in here, so we can try. If we are interfering with this,
It will open.
we are blocking X. So what would happen to the following ions? Sodium, calcium, potassium, and the membrane potential. So firstly, tell me what's X, Y, transporters. What are they? Yep. What's your name?
جميل بس كيف يعني
جميل.
شو الاكس واي بالضبط؟
XY. So what is this type of transporter? What is this type?
انا ناسي بس انا
Transporting potassium against its concentration gradient, and sodium is being transported against its concentration gradient, right?
yes.
So what would be X? What type of transport is this?
active.
Active, right? X is a primary active transport. You got the same question TBL, right? So X is a primary active transport. And Y? What is Y? So sodium is moving down its concentration gradient, while calcium is being transported outside against its concentration gradient. So what is Y?
Antiport, it's something
It's an antiport, right? It is an exchanger because the directions are opposite, right? Now, if we block X, if a drug block X, the primary active transport, what would happen to the cell? What would happen to these ions?
So basically it will increase.
Sodium will increase.
Yes.
Okay, so increase.
And potassium will decrease because it's hot.
Okay, so let me see. So potassium would be decrease.
Potassium will be increased because it's and the membrane potential is depolarized because the Na comes in.
Depolarize. Okay. You guys agree with him? Why wouldn't you agree? What is not working, then the exchanger wouldn't be working as well. And so calcium would be moving down its concentration gradient and will make the concentrations of calcium inside higher, okay? And this will make the membrane depolarized, more positive, okay? Is this all clear? Okay.
This all found in the previous question.
Okay. So, yeah. So this is the end of the session. If anyone has any question? Yes? Which one? Number 14? Yep. Yes. Tonicity describes the effect on the cell volume. Yeah. Yes, because we have injected water, which made it hypotonic. Hypo is smaller. I don't understand. Osmotic, yeah. Yes. Yes, so yeah, the patient red blood cells will swell because the blood fluid is hypotonic. Yeah, you are right. The words of the questions are not accurate. Okay, but the concept here is that the fluid itself is hypotonic and it will make the cell swell. Okay, you see? Because sometimes some of the words, you know.
دكتور بس عندي سؤال عن الفاسيلتيتد diffusion question.
Yeah, which one?
اللي هو
Yeah. Let's let me take the Yasser's question. I'll come to you just for a second.
yes, how is number 9 E?
Because number 9, the movement of glucose. So Yasser is asking why number 9 is E. Why is it E? Okay. Why is it E?
It's supposed to be D, symport, no antiport.
Okay. So it's a co-transport. Yeah, you are right. It is a co-transporter.
T.
So it's the type. Yeah. Okay, your question? What's your name?
I have a question about the last part.
Okay, so what happens is that when the active primary transport is blocked, this will also affect the secondary active transport, right? Because the secondary active transport is dependent on the primary active transport. And so in this case, ions will move down their concentration gradients. So sodium will move from area of high concentrations from outside to inside, will make inside more, okay, higher concentrations of sodium. Potassium is the opposite, it would move to the outside, okay, and so it will be less. Calcium is higher in the outside, as you see in the question, and so it will move from outside to the inside, okay? So it will make it have higher concentrations of calcium, okay? Yeah.
So you said that over here the primary active transport and the secondary are dependent. So if this was affected, would this be affected by this? Thank you.
Okay, so this is the end of the session. I hope you all do great in your exam and please study well. Okay, this is not fully representative of the exam. Study very hard. This is just an exercise, okay? And I wish you the best in your studies, okay? All the best. Yep. If anyone has a question, come to you.
And this question, if we do the neuronistic question, we have them. If we do the V